Thank you, and thank you to the conference organizers for accepting this abstract for platform presentation. I'd like to acknowledge first that many of the analyses I'm going to share with you today were done by Sarab Sihag, who couldn't join us, and he was an intern in my lab for 2 summers from Rensselaer Polytechnic Institute, where he's completing his PhD in electrical engineering and in the middle of his qualifying exam, so he sent me. The objective for this study was to assess the appropriateness of what's called graph signal processing, or GSP, for fusing diffusion and functional magnetic resonance imaging. So one is a structural measure of brain connectivity, and the other is a functional measure of the bold signal. These were recorded from patients with a history of concussion, and the outcome, I'll tell you up front, was that we found that structure-function biomarkers derived from GSP are appropriate for categorizing post-concussion syndrome patients from healthy controls. But what we also found, which is interesting, is it correlates very well with serum or blood neurofilament light chain proteins. So the search for biomarkers in neurological outcomes regarding concussion and post-concussion syndrome is necessary in order to aid in the design of new therapies and to mitigate the risk of post-concussion syndrome, which we are interested in. The neurodegenerative properties of this trajectory require clinical biomarkers that can tell us more about neuronal, synaptic, and tissue modifications and degeneration. Now, NFL, as we've heard in talks from Dr. Smith and others, is released by axonal damage, and it's elevated in really a plethora of different neurodegenerative diseases, including TBI. And it may become, and is becoming, a key biomarker for individuals at risk. So we wondered if applying these new graph signal processing techniques that come out of the engineering field could be appropriate for finding these multimodal biomarkers, and not only to help with identifying patients at risk, but to maybe explain some of the underlying, at least present hypotheses as to the underlying mechanisms by which these blood biomarkers may be coming into the patient. So to begin with, I'll describe the data inputs. We used dMRI tractograph data, basically the tracing of diffusion tensor imaging from one cortical area delineated by the Desikan-Kiliani atlas to another, and that's represented by a matrix of 1s and 0s, which basically describes which areas are connected to which. We also used fMRI bold signals recorded from the same patients. These are fluctuations in each of these brain areas in basically metabolic activity and neuronal activity. And we also used serum NFL levels from these same patients in healthy controls. And these were all shared by the lab of Carmela Tartaglia and the University Health Network. We had 21 healthy control subjects and 37 former athletes. Graph signal processing is the method we chose because brains recovering from TBI, and whether mild or otherwise, they exhibit dynamic changes, both to the functional and to the structural networks of the brain. And so this is an ideal analytical technique for fusing the two types of networks and to derive features from them. So what we do is take a graph such as this, represented by the matrix, and we decompose it spectrally. Okay, so this sounds a little intimidating, but what we do is project that onto the surface of cortex, and you can see that high-frequency harmonics of the connectome have variation across the brain, which is faster than the low-frequency harmonics. And a simple analogy as to how filters work is the atmosphere of the Earth. So when lightning strikes, if you're far away, the Earth's atmosphere is a low-pass filter, so you hear a muffled, rumbling sound. But if the lightning's very close, you hear all the high-frequency energy, and it's very startling, and you hear a crack. So similarly, this low-frequency energy is muffled and slowly varying across the brain, and this high-frequency energy in the connectome of the patient itself is varying more quickly. And this was described in a paper by Adesoy et al., where some of these harmonics are actually correlated to the default mode network. So it gets a little more complicated when we add fMRI to the problem, and GSP actually asks the question, not just what is the connectome telling us through these harmonics, but what happens when we construct a filter from these harmonics? Okay, so it's a graph filter. In this case, it's high-pass. High-pass filter, much like a filter that you would apply to a signal, such as a time series signal. And what you get is a very non-correlated set of fluctuations in the bold signal, which is varying very quickly across the brain. The low-pass filter is much like the thunder analogy, And that low-pass filter is also derived from the graph. And you see that there's a lot of correlation and structure in the way things are varying across the brain. And in the initial paper in IEEE describing this method, Huang et al. Said that GSP measures variability across regions of the brain for a fixed time, a sort of spatial variability with respect to the connectivity pattern. So it's a little complicated, but think of a lightning strike and not just filtering it through the atmosphere, which is a very uniform medium to construct this low-pass filter, but instead filtering it through some convoluted network of pipes, such as a subway system or a sewer. That would give a very different type of filter property. And so that's one analogy that might help you understand how these two structure and function connectivity traits are being fused through GSP. Okay, and so what we do is take these signals and measure the area under the curve, the L1 norm. That becomes a feature. For each brain area, for each subject, we measure the high-frequency energy and the low-frequency energy coming out of the GSP filters, and much like a microarray, we do statistics. So what we found is that the left hemisphere high-frequency energy from patients and normal controls gave us 5 brain areas which were very good at separating the two populations. So that was interesting, not in the right. Whereas the low-frequency energy coming out of these filters gave us four brain areas for each, basically, the left and the right hemisphere. And these were significant because the effect size was greater than 0.25. So that was a very interesting result. Even though we have a multi-sampling problem, we found these areas to be sort of significant beyond expectation. And that would mean that the areas need further examination. And that was how we did the preliminary categorization between PCS and normal healthy control patients. And I want to point out that some of these brain areas that were picked up, especially in the high-frequency energy coming out of the GSP filters, are implicated in post-concussive syndrome, not surprisingly. So left entorhinal in terms of gray matter disruption, precuneus in terms of disruption to the default mode network, and superior parietal and cingulate in terms of white matter disruption. So there's many things that are consistent between biomarkers or features from the brain imaging data that have been done in a unimodal type of study, and this now multimodal study that fuses functional and structural connectivity. The low graph frequencies also had, like I said, 8 different areas which were good at separating the patients, and these came from both the left and the right hemisphere, as I've already mentioned. Okay, so what did we do next? Well, we had some data that was which was quantifying patients' blood or serum NFL levels, and so we added the additional task of taking these energies from the high frequency and the low frequency filtered signals, filtered on the patient's own connectome subject-specific data, and regressing it, or performing what's called a partial least squared regression. Basically, you take a whole lot of features and you try to predict a specific feature, in this case, serum NFL levels. And now, why would we do that? We think that the effects that are being observed in serum NFL are both relevant to disease, but there's also quite a bit of variance in the healthy control population. So we began this study with doing this regression against just the healthy controls. And here's what we found. So initially, we did the naive thing and regressed all 133 features, 33 different brain areas times 2 hemispheres times high and low plus age, and we found that we could predict 100% of the variance, and that's not true. This is basically an artifact of what's called overfitting. Okay? So we took an approach which is very well established in the field, which was to use what's called the variable importance projection of each of these features. So these are the measures of variable importance projection for each brain area for the high graph frequency and the low graph frequency energies. And we took a threshold, and we found that at that particular threshold, instead of being insignificant with compared to a randomly shuffled distribution, we can get at 1.4 a significant explained and predicted variance. And it was quite significant in that with this model, which now is represented in terms of which brain areas survived that filtering plus age, we were able to predict serum NFL levels very well with 77% of the variance explained. This is surprising given that age alone in the healthy control population, the thing that is most sort of commonly associated with variation in healthy controls, only explains 29% of the variance. And similarly, our predicted variance with this model is 54%, while age alone explains just, or predicts just 19%. Now, some things that were not sort of baked in that we wouldn't have expected and are surprised by is that the high frequency energies in the brain have a positive correlation with blood NFL in healthy controls, whereas the low frequency energies have a negative correlation. So that's surprising. For the former athlete population, we also performed the regression, and we found that we construct a different model from that data. It's a disjoint set of features. That's also a little surprising, but you'll see it's not quite as surprising as you might think, in that these are probably correlated with the other brain areas that were used to construct the healthy control population model. And again, 65% of the variance is explained, 49% of the variance is predicted, which is very good compared to age alone. So the last figure I'll show you is what happens when we take the former athlete data and we try to predict blood serum NFL levels using the former athlete model, okay, as opposed to using the healthy control model. So again, this is just a regressor, much like y equals mx plus b. So it's a simple regression, but it's in many dimensions with these 7 or 12 different variables. And what we found is that we can use both models to predict the serum NFL levels. So it's not that the patients and the healthy controls are that different, it's just that they have different features in their model. But one thing we found interesting, and I'll close with this, is that for those patients who were former athletes but were better predicted by the healthy control model, okay, those are places where this green line is closer to the real data in blue than the red line. You see those are marked by a star. These were all among the former athletes who had sort of, on average, higher NFL levels than the rest of former athletes. So that's mysterious. We think this is a good opportunity to take this method and move forward. It not only explains much of the variance in the healthy control population, but it presents opportunities for new types of biomarkers in the actual graph signal process data. So we applied this to brain regions, and we found a good and appropriate use for multimodal fusion of brain imaging data in the construction of BCS biomarkers and predicting blood biomarkers in serum NFL. Thank you.
So, that now it's becoming, a political issue to say PCS, and especially because of headache and other things. So did you parse out what PCS was in this case?
No. We used the data that was categorized by Carmela's lab, and there was a host of other cognitive measures and other sort of ways in which these patients were categorized, which we are now analyzing in collaboration. Yeah.
These are former professional athletes who've had multiple concussions. Many of them have complaints, and none of them are demented. Those people have been excluded. We've ruled out Alzheimer's disease in them. So headache is not a big feature, but they have cognitive and mood complaints. But they're not actively post-concussion syndrome. The last concussion in these people is years, if not decades ago.
This is such a naive question that I'm hesitant to ask it, but does this indicate the value of doing DTI or of doing NFL levels or both?
I'm glad you asked it, because it gives me a chance to take a little bit more of your time and explain what motivates sort of us at IBM in getting interested in this type of work. The hypothesis we're exploring is that the brain activity and modifications to homeostasis in terms of network dynamics, how the brain sort of regulates itself, is disturbed in TBI and neurodegenerative disease, and that is a necessary input to our understanding of the pathophysiology. So this is only one way to explain the result. The other way is that this is just a correlative aging, and it's correlating with other progressions that are going on in the molecular pathology of these diseases. But if the former is true, then yes, the answer to your question is yes. We need to take very seriously that just as the Y chromosome causes concussion through the behavior of the individual who has that gene, that the brain's behavior needs to be taken into account as a causal link in terms of how an insult to a set of axons and diffuse axonal injury in the splenium of the corpus callosum can lead to changes in the molecular pathology in areas that are distal from that initial injury. And that could be due to changes in the relationship between brain metabolism as measured through the bold signal, coupled to the actual network that the patient brings to the problem of sustaining brain activity, and that itself could be a way to kind of additionally reason about changes that are going on at the genetic and molecular level.